Um, well, welcome and welcome back to our sessions on model organisms. And there was one of them right there. Our speaker today is Charles Baer, Dr. Baer, who is a professor of biology. And he seems awfully loud, but all right. He came to the University of Florida in 2003, so he's been here 20 years. He is a professor of biology. Before coming here, he was at a postdocs at the University of Colorado, the University of Oregon, and Indiana University as well. So 3 postdocs, and then after having completed a PhD at Florida State University. And then he came to, and then he came to the University of Florida. So Dr. Baer is an evolutionary biologist who is particularly interested in mutations, the sort of origins of variation, and in particular, he is interested in the fact that there is quite a lot of variation in the rate of mutation in different genes and in different organisms, which is quite a puzzle, an evolutionary puzzle. So he has been working on that for many years. But today, he's going to tell us about this creature here, which is the model organism that he's been working on, Xenorhabditis elegans. And if you are here, you could take a look at, he's brought some of these creatures, and you can see how small they really are. They're in this microscope up in the front. Okay, so Charlie,
all right,
take it away.
Thank you, Jane. And let's see, back, forward, and L. This must be the light laser. Yeah, okay. So this is just a video of C. elegans that like hit toward the top of my Google search for videos of C. elegans. I didn't actually take it myself. Like I said, I brought up a couple of plates of C. elegans where after the show you can come up and take a look at them. I should have brought a pick so I can show you how we pick them off because we use like a little platinum pick, or some people use eyelashes to pick. I've never actually used an eyelash pick, but so the first time I ever picked a worm, it took me like 20 minutes to pick the worm. I pounded half a dozen of them into the turf, and I was supposed, like the next week or 2, I was supposed to start an experiment with 600 samples. And I was like, man, if it takes me 600, or if it takes me 20 minutes to worm, this isn't going to work. But I got faster. So, yeah. Okay, so that's this is a picture we took. Here they are in. Those are in a liquid and not on a plate. Okay, so let's see. There we go. Okay, so I was going to. Well, I guess Jane introduced me, but I can tell you a little bit more. Then I was going to introduce the worm and tell you a little bit of the history of C. elegans. Well, the history of C. elegans as a model system, and then 5. So whoops, back. Delays, yeah. So, spend probably most of my time talking about worms as a model for developmental biology. Also, neurobiology is intimately involved with the development and glowworms and the worm genomics and RNA interference, which probably many of you have heard of. And finally, something that I think is especially interesting, transgenerational inheritance. And I will also say that, giving a talk with Dr. Burns in the audience is in front of him. Oh, sorry. Okay. There you go. Okay. So, all right. There you go. Whoops. That's the laser. All right. Forward.
This video provides a brief introduction to a very popular model organism in developmental biology. The roundworm Xenorhabditis elegans,
hitting
or
forward.
C. elegans for short.
There. Okay. All right.
Let me
back.
start out with a little bit of background information.
Let me go.
C. elegans.
Oh, okay. So
roundworms.
but first, a little bit about me. So this is the Baer research program, circa age 9, and I was, one of those kids who liked to look under rocks and logs for salamanders. And I bet a lot of people in biology say, well, how come you're a biologist and not something else? Well, I like to look for salamanders when I was a kid or whatever. I like to fish and hunt and run around the woods. And so eventually I wound up as a master's student at the University of Texas along Mayett Wave. And my plan was, all right, I'm going to get a master's degree in biology, and I'm going to go to work for Texas Fish and Wildlife. And I'm going to drive around a 4-wheel drive truck, and I'm going to ride around on a boat, and I'm going to, marry the girl at the donut store, and I'm going to have a nice life. And then I wound up in the Department of Zoology at the University of Texas. They had all these great scientists, guys who forgot way more about science than I'll ever know. And I was so naive and so unintimidated by it. I'm like, you're a college professor. How the hell hard can that be, right? So at some point, my ego kicked in. I said, hey, man, if these guys can do it, I can do it. And I want to sit at the grown-ups table. So, 30-something years later, here I am. And like Jane said, also, I use C. elegans as my model organism. For me, they're just little meat computers. And I will, actually, I guess, but before I get to that, I want to tell you. Okay, we'll get. Okay, so how did C. elegans come to be a model system in biology? And this goes back to the early 1960s, and Sidney Brenner was one of the early big names in molecular biology, and he worked on bacteriophage and also E. coli, and I can't remember what his signature contribution was. I should have looked that up. But so this is early in 1963. He writes to Max Perutz, who's the director of the MRC, the Laboratory of Molecular Biology, the house that Crick built, right, at Cambridge, and he, somewhat arrogantly says that, nearly all the classical problems of molecular biology have either been solved or will be solved in the next decade. So I assume what he meant by the classical problems is DNA replication, transcription, and translation. And said, okay, now we're going to need to figure out, what's going on at a higher level. So the future of molecular biology lies in the extension of research to other areas of biology, notably development and the nervous system. So he set about, so he proposed, he's looking, okay, what would be a good organism to address these kinds of questions? So he proposed, I was looking at several model systems, so small metazoa. Metazoa is just a fancy name for animals. It took me years to learn that. Chosen because they would be suitable for rapid genetic and biochemical analysis, which is what he had been doing in bacteriophage and bacteria. So the criteria were it had to have a rapid life cycle, so you could, genetics, from after genetics, you have to get parents and offspring, and F2s and F3s. So the faster the life cycle, the faster you can do your experiments, and you can get a large population of them. So large sample sizes are good. A simple reproductive cycle in genome for tractable genetics. And there's a lot of small organisms that do not have simple life cycles. They may be facultatively sexual or asexual, or they may have, think about malaria parasites, you have to go through multiple different hosts. So they had to have a simple reproductive cycle and not a ridiculously large genome and small size. And if you saw those of you who did look at the worms, they're small. A whopping big hermaphrodite is about 1 millimeter long. You can barely see them with the naked eye, but you can. So he got a sample of C. elegans from E.C. Daughtry, who was an early fan of C. elegans. Said, man, you ought to work on this. And he said, okay, these are pretty good. So here, this represents the life cycle of C. elegans. They're antrodioecious, and that means there are males and hermaphrodites. So in a typical culture of C. elegans in the lab, there are more than 99% hermaphrodites. The hermaphrodites are self-fertile, or they can be fertilized by the males, which occur at less than 1% typically in a lab population. But one hermaphrodite cannot fertilize another hermaphrodite. Has to mate either with itself and self-fertilize, or with a male. So they have what's called an XO sex determination system. So the hermaphrodites have 2 X chromosomes, and males have 1 X chromosome. And the males are generated spontaneously at less than 1% by non-disjunction of the X. Now there are wild strains of C. elegans where you get many more than 0.1% males. Most strains, sex ratio is about. So they had at 20 degrees, they have about a 3.5-day generation time. At 25 degrees, they have a 3-day generation time. If you want to get a generation time faster than that, you have to work with microbes, with yeast. So this is about as fast a life cycle as you can get for a multicellular organism. They are bactivorous, so I have them on a culture of E. coli. So we just spot up, we got an agar plate, we put, a spot of 5 microliters of overnight culture of E. coli, and worms are fine. So super, super easy to keep in the lab. And there is so here's the life. So here you have a hermaphrodite adult. She lays her eggs. They hatch after 12 hours or so into L1 stage larva, which molts after another 12 hours, or an L2. And after about 3.5 days, we're back to here. So if the conditions are stressful, if they starve, or if they're at high temperature, or whatever else is stressful to the worms, the L2 will form one of these, what's called a Dower stage. And basically their mouth, they like grow a plug over their mouth so they can't eat. And these guys are really stress resistant, and I can have a plate sit that I've forgotten on the lab bench or in the incubator for months. And I get it out as Brad Barbazuk, Brad used to work on worms before he worked on plants. He calls a dried up plate of potato chip. And I've gotten worms off of potato. Great. So these guys are wonderful lab organisms. Also, and ask any worm biologist in the world, why are worms so great compared to Drosophila, which you heard about a couple from my lovely wife, or anything else? You can cryopreserve them really easily. You can freeze them down. I've thawed out worms that I froze down 20 years ago. And so, you can take a vacation. So yeah, I'm going to go fishing for a week, and I'm going to put my worms, I'll freeze my worms down if I have to, because I could just let them devour. But this is great. So, I mean, if for no other reason, so what in the experiments I do, which maybe I'll talk about a little bit later, I can, if you're doing an evolution experiment, I'm an evolutionary biologist, I can, grow up, I can take a plate of worms and I make a few replicates, and now I grow this culture up, freeze it down, so I can compare the evolved descendants directly to their unevolved ancestor. And you can't do that with flies. You have to keep a big cage of flies and hope they don't evolve or get contaminated. So it is this ability to easily cryopreserve the worms that is by far the best reason to use worms. So you can synchronize a culture, and if you're a developmental biologist, you say, okay, I want to work at this stage of development. They're really easy to synchronize because we do what's called bleaching. We grow up a culture of worms, and we soak them in dilute bleach solution, and the eggs that you saw here, right? So the embryo, it's got like a waxy coat on it. And they can, if you put the adult in bleach, it thrashes for a bit and then it pops and eggs spill out. But those eggs can last for at least a few minutes. So now, the worm only has so many eggs in her. So now you have a plate full of eggs. They all hatch out. All those worms are the same age within probably a few minutes to an hour or so of each other. So you can get a large population that's very closely synchronized at the same stage of development. So that's a real advantage too. So because they are self-fertile and because males are so rare, they exist in nature primarily as nearly homozygous strains. So this is good because if you're interested in doing genetics, it helps to be able to have an isogenic line, so you get multiple individuals with the exact same genotype. If you do this in flies or something, you have to inbreed them, and respectable organisms like flies or humans or whatever really, really don't like to inbreed, so you have to go to tremendous effort. There are other species of worms, Cinerobditis, that are respectable male-female species. They really, really do not like to inbreed, and you have to really work hard to get an inbred line of outcrossing worms. Not a problem in C. elegans. Okay, also, as you probably saw from the video, and you can see if you go up and look at the microscope, the individual worms are transparent. So if you are interested in what's going on inside the organ, I can't look and see Ken's liver there. I have to sort of kind of guess at what's going on in Ken's liver, whereas if Ken was a worm, I could just sort of focus the microscope and say, whoa, here's what's going on, right? So the transparency for the purposes of microscopy is seeing what's going on inside the animal. Okay, so those are the many reasons why Sidney Brenner et al. Hit on C. elegans as a great model system. Okay, so this is the abstract from probably the first important C. elegans paper. This is a single author paper by Sidney Brenner in 1974. So let's see, methods are described: isolation, complementation, and mapping of mutants. So they found about 300 mutants. So they were doing a mutant screen. I've never done a mutant screen, and a mutant screen seems like magic to me. But what they do is you pour a mutagen on a plate full of those worms, and then you plate them out, and then you look for something weird. Right? And if you see a worm doing something weird, you say, that's a mutant. And having a completely inbred strain, you don't have to worry about any background genetic variation. This is a new mutation. And, they did this Brenner and his lab did this painstakingly. And I don't think anymore, if you found like 4 or 5 mutants, you'd probably write the paper. But back then you had the freedom to, hold off for a few years because they started this in, I think he started this this may have been like 10 years, almost 10 years work. But they found 300 mutants and something like 100 genes. And mutations in 77 of those genes alter the movement of the animal. So he's looking at the worms and says, this worm's doing something funny. Right, so this is, this is a tremendous amount of work. But Brenner 74, the seminal worm paper. Just like with the nervous system, that's a pretty small number of overall cells in a pretty simple. Okay, so as I mentioned in, I think the first quote, they were especially interested, Brenner and his colleagues were especially interested in development. So this is later on in 1963, Brenner writes to his boss, Max Brutz, and says, here's a proposal for what we're going to do for next however long. We propose to identify every cell in the worm and trace lineages. We shall also investigate the constancy of development and study its genetic control by looking for mutants. So for the sake of comparison, so I can cut to the chase here. The worms have 959 somatic cells. So that's a pretty useful trait. And I have about 1 trillion.
what's known as an invariant cell.
And yet, you know, with just if I squint really hard, I can see one of those worms with my naked
So I'm
eye.
not going to go into detail
Like,
on this right now.
so you know, I mean,
Suffice it to say
so
that
what
this
you know
means
what
is
can be done and
that
what they
all the
did is they sort of
959
work.
somatic
First, they
cells
they
are
worked their
formed in
way
exactly the same
forward,
way from one
looking to see,
organism
okay, a
to
cell
another.
divides
In other words,
here,
the specific
and
cell
this cell
structures
divides
that
here,
occur
and they
in the
started
adult
out.
organism, those are being formed at the same
So I should say, this is
same
John
place in a
Sulston,
very reproducible
and this
fashion.
is Bob Horvitz, and those guys won a Nobel Prize
Together with the simple
for
anatomy,
basically for the
overall
worm, but
cells
really it
that
was
make up
for
the adult
working out
anatomy and
the
the transparent
cell by
features
cell
of
development
embryos,
of the worm.
that
And that
makes
was
it
in
really, or
2002,
makes it
I think.
relatively easy to follow
So,
processes in development.
okay, so I mean, obviously, I don't expect you to read this. So this paper is this was 77, Developmental Biology 77. This is the first paper by Sulston and Horvitz on post-embryonic cell lineages of C. elegans. They started with a, one of those.
Those are rare in the
Go
wild,
back here.
but they can be maintained by crossing in the lab. So the hermaphrodites can cross-fertilize like a female as well as.
So they started with an L1
just
hatched out,
progeny,
and they followed every cell in that in the developing worm until they got to here.
They're expelled into the environment through an opening in the body wall. So they complete development
So,
internally.
and here I've highlighted, we have determined these cell lineages by direct observation of the divisions, migrations, and deaths of individual cells in living nematodes. So they looked at a lot of worms, and these were genetically identical. It's one strain, so the laboratory strain, the N2 strain of C. elegans. There's a famous picture out there. So John Sulston was the one who did most of the microscopy. So he had his microscope station here, and he had something over here. Maybe it was his note. I don't know what, he had something over here. And he wore a groove, and he was sitting on a chair with wheels in it. And this is in his lab in Cambridge. And he wore a groove in the floor with the wheels of his chair moving from the scope over to here to whether he's writing, I don't know, doing something, right? I forget what he was doing. But he looked at an awful lot of worms to figure out this. So this is an image, a figure from that paper, right? So I didn't show. So basically, it starts at time 0 and goes through to an hour, right? So he's looking at these. This is one worm, and this P9 cell here, and the P10 cell here, and you can see the nucleus of the P9 cell. And here it's, the P10 is starting to divide, and a little bit, 30, this is 26 minutes, 34 minutes. So P10 has divided now into the P10A lineage and the P10P lineage. And by an hour now you have both the P9 and cell and the P10 cell have both divided. So these 4 cells here at this time are descended from these 2 cells an hour earlier. And this is just, this is just 2 into 4. They start at the L1 stage. I think there's something like 500 cells that divide out to 959. So, I mean, it just boggles my mind that somebody could have the patience to do that kind of work. So, okay, so this is the next big paper, Sulston, Schoenberg, White, and Thompson. So this was 1983. Now they took it all the way back to the beginning. So the first paper, the Sulston and Horvitz paper, was just the post hatching to adulthood. Now they started from the zygote right at the beginning. So the P0, this is the zygote, right? So at the 2-celled embryo, you get what they call the AB cell and the P1 cell. And AB goes on to develop into those things. The P1 then, so here's what it looks like. Here's the P, it divides into the AB cell and the P1 cell. And then here, P1 now divides into the EMS.
But it's also
In the
really
P2,
and inexpensive
AB has
to
divided
lower
into
numbers of
ABA.
C. elegans in the
And
lab.
then
And this is another
EMS divides
advantage
into
of
the
model
MS and the
So
E.
it's related
These go
partly
on to
to,
form
their
those
small
things.
size
And
life
this,
large food
let's
sizes,
see,
which
the
we've already talked
P3
about,
divides into the
but also
P4,
conditions
which goes on to
used
form the
to
germline,
obtain them in the lab.
forms the gametes.
So you can
So
take
there are
a
1,
little
2,
plate like
3,
this,
4
use in
divisions
microbiology,
before
and
now
you
the
can see
germline
surface with,
is
some E. coli
dedicated.
bacteria, which
So
are
the,
ones we
another
use
interesting thing, and going back to the
And
idea
then
that
if you let
what
that grow
what
for a couple of
Horvath,
days,
or Horvath,
you'll have an
Brenner
artificial
was
this
especially
film
interested
of E.
in
coli
was
growing on the surface.
neurobiology and the
And then you use a
neurodevelopment.
tiny metal wire
So
called a pick
neurons
transfer some hermaphrodites
are
to
the
the
AB.
dish.
Some
E.
neurons
coli eating it
are traced from the
generating
AB
lots and
cell.
lots of progeny through
Other
some
neurons are traced from the
So
MS
after a few days,
cell,
you'll
which
have a
is
plate full of hermaphrodites. So this is what they
which is
look like
here,
under a
and
nice
other neurons still are
as they swim around
derived
on
from the
the
C
plate in the E. coli.
cell here.
You
So
see various
it's not like so
here, along
for
with
whatever
some
reason,
hermaphrodites and some
this lineage can now, so the same thing with
So
muscle.
as
Here we got
E.
muscle from
coli or
MS and
coli,
muscle from C.
you just transfer some of them to
And
green
D,
plate.
intestine is only from the
There are
E
lots
and
of
germline.
ways.
So
You
there's
can
some
expand
some
your lab population very, very
these
quickly.
lineages have not completely differentiated
Final
yet.
thing I want to emphasize in terms of advantages of
So I mean,
working with
the
C.
punchline
elegans
here is they
organism is
over
that there are some really great
this was
experiments
83, 73, over 15 years.
C. elegans development.
These guys worked out
Now, I'm
the
going to tell you a
entire
bit more
fate
about,
map
the
of
details of these techniques
an
a
organism.
little later in
And
this
as
unit.
far as I know, that
More
hasn't,
specifically
that's still
through
C.
the
elegans,
tools, that we
maybe
use
some
in
related
So
species
first
like
of
C.
all, I
briggsii are
already
the only
mentioned that C. elegans
multicellular
are
organisms that we have a
amenable to
complete fate
genetic
map for.
screenings.
I'm pretty sure that's true. Okay, so here it is, right? So
So,
959 somatic cells, and this is here's the P0, the zygote, the AB cell, and the
so that's one
E
important tool.
EMS, right, and the MS lineage, the
has
EUC,
a
et cetera,
genome.
right? And notice also, some of there's also some of those cells die off by apoptosis, and some lineages divide a lot more than others. The 959 somatic cells, of which there are 302. 302 are neurons. I think there's 59, whatever they call those neural support cells. There's of those 302 neurons, there's 118 morphologically distinct classes of neurons. And of all of our trillion cells, probably a couple 100 billion are in our brain. So, and a worm has only 302. And I'm not going to talk. I mean, I don't do anything like neurobiology, but you can ablate any one cell with a laser. You can destroy that. So anywhere in the lineage, and you can sort of say, okay, what happens if I ablate? I don't know, if I ablate this neuron, what does that do to the worm's behavior? Now it only turns left or something. So it's straightforward to map individual neurons to behaviors. Also, there's a lot, they've worked out lots and lots of genetics to where if you knock out a particular gene, one neuron will develop into some other neuron or something like that. Okay. Okay, so that was the first story. So feel free to ask questions at any point. That was the yes, Len.
In these really cool time-lapse movies. Now, this adult hermaphrodite is expressing two different fluorescent proteins. One of them is tagged with green fluorescent protein.
Dr. Baer, can you repeat the question for those on Zoom?
So the question is, were there any surprises? Were there things that people really, and I guess I would say almost surely there were, but there's none that were so surprising that they have filtered down to me. put it that way. But I think you wouldn't necessarily have predicted. You might have thought that all the neurons were derived from one lineage early on, or something. There's lots of things that could easily have gone otherwise. I can't think of any.
You just had to close this. It was playing in the background. Sorry.
Oh, okay. So I mean, probably, but I don't have a. Oh yeah, by the way, here's what it is. So, okay, so here's the second story. So, if you go to PetSmart or something, you can buy. You guys know glowfish, like they got the fluorescent fish that have fluorescent green or fluorescent red or blue or yellow or what have you, right? So what those fish are is they carry a transgene. They have had a fluorescent protein, a gene that encodes a fluorescent protein engineered into their genome. So now they stably express fluorescent protein. That was so. This guy here, Marty Chalfie, and two other guys won a Nobel Prize. I think that was 2004, 2005 for GFP, green fluorescent protein, which is a naturally occurring protein that's found in a jellyfish, Aquaria victoria. And so Chalfie was in a seminar and heard some biochemists talking about this fluorescent protein. And he's, thought, hey, I wonder if you could engineer this into a worm. So he went back to the lab and he had a graduate student doing a rotation in his lab. And he said, hey, I got a cool project you can spend the next 2 months working on. So this woman whose name I can't remember, but you can look it up. It's in his Nobel lecture and everything, managed to clone or get this this green fluorescent protein gene cloned and into bacteria. And then she left the lab to go on and do something else, and he turned it over to his technician, and his technician managed to clone the GFP gene, whoops, let me go back here, into C. elegans. So the first multicellular organism to have a fluorescent reporter gene integrated into its genome was C. elegans. And this is the cover of Science in February 1994. So I say this earned Chalfie and his two other guys a Nobel Prize in chemistry. So this is just like a little this is actually, I think it's taken from his Nobel synopsis, I guess. So basically what you would do, so a promoter, this is just the part of the gene sequence that tells the transcriptional machinery, okay, start transcribing this gene. This would be the start codon for the protein, and this is the stop codon, the 3' untranslated region. So if you put this into a worm, and there's various ways to make transgenic worms. You inject it in, or you can use a gene gun, and now you still have to, if you use CRISPR, you still have to inject it in. I've never injected worms myself. My colleague Keith Cho does that all the time, makes transgenic worms. So if you do this, then you are just expressing GFP, and whatever, if you say, okay, I want to express it in, this is muscle here, right? I will put a promoter for myosin, which is expressed in every muscle. So wherever you have myosin expressed with a myosin promoter, it'll glow green. Or if you have some other gene and you want some other protein, you say, I wonder where this protein is expressed, right? You put your promoter that's going to express this GF, and then you make this construct that has your unknown gene linked up to GFP, and wherever in the organism it glows green, you say, aha, that is where my unknown gene is expressed. Or you can do this, and you can ask what happens. So here you've put, what have I done here? This is one, if you want to know what happens when you break the gene, I think. Now you put a broken gene in, and you see what happens. You say it's expressed here, and if I break this gene, then the organism stands on its head or something. So there's all sorts of tricks you can do for learning functional biology, and the key here is having the reporter that you can, again, and having a having a translucent organism really helps, because even if Ken's liver is glowing green, I can't see it. That makes sense? So this is also from the Chalfie Nobel synopsis. So here is, these are various green fluorescent organisms that people have made, and I'm sure this is not an exhaustive set. Glowing green mice, that's pretty cool. It beats, I don't know, it's coral or something down here. And coming back to the theme of neurobiology, this represents, like, these are all the neurons in the worm, right? Here's the head, here's the pharynx where it's running up through. This is one of these is the ventral nerve cord, and the other one is the dorsal nerve cord. I don't know which. And then there's these cross connections between ventral and dorsal. And then here's the anus. And here's the vulva, which is the hole in the side of the hermaphrodite, in which the male will mate with her and she spews out eggs. So if you want to look at, okay, what happens if I ablate this connection, then what happens, right? It's really easy to look and make sure you've done it right, and get the picture and what's different. So, I mean, so fluorescent reporter genes are hugely important, and it's sort of it's sort of hard to imagine how people really did this kind of biology before having fluorescent reporters. All right. I know I got more stuff than there. Okay. The worm genome. How am I doing for time? I have no idea.
It's 2:08 and we have until 2:30.
Okay. All right. So I'm doing pretty good. All right. So as surely you all know, the Human Genome Project was a big deal. And it was launched 84, 85 or something like that. And it was officially kicked off, and which the money was signed off, I suppose, in 1990. So the human genome is about 3 billion bases, and the worm genome is about a 30th of that. It's 100.15 megabases. So shortly after the Human Genome Project was conceived, again, John Sulston, and this is Bob Waterston, talked the NIH powers that be into funding the Worm Genome Project. It's sort of a pilot project for the human genome. And the complete, I have in quotation marks, but the pretty complete, the complete-ish worm genome was published in 1998. So 6 years between the time they kicked it off and the time the paper appeared in Science, I think. So C. elegans was the first multicellular organism to have its genome completely sequenced, and we are very proud of that. So some natural history of the worm genome. So there are about 20,000 protein coding genes in the worm genome. People will argue about how many coding genes there are in the human genome, but 22,500 is a number you hear somewhere between 20 and 30. But it's on the same, very close to the same number of protein coding loci that are in the worm genome. Those guys have 959 cells, and we have 1 trillion. So and yet, the plans for the proteins of the organism are, you might have thought, well, we're way more complicated in some sense. We must have a lot more genes, and yet we don't. So there's about more than 20,000 non-coding RNA genes in the worm genome, most of which are small RNAs, and there's various kinds of small RNAs, and we're learning more about the small RNAs every day. The number of non-coding RNA genes in the human genome is completely unclear. Maybe also on the same order of 20,000, maybe more, but I don't think we really know. So about a quarter of the worm genome is protein coding. In contrast, about 1.5% or so of the human genome is protein coding. So 20,000 or so genes in each. So, whereas a quarter of which of the worm genome is protein coding and less than 2% human genome, what else is in the human genome? Well, it's mostly filled up with transposable elements and other maybe junk, maybe not. So worm 6 chromosomes, 5 autosomes in the X chromosome. So here's what I think, from my perspective as an evolutionary geneticist, that's really interesting about worms. So worm chromosomes, they have the center. So worm chromosomes can be divided up into centers and arms. And the centers are gene rich. Or most of the worm's genes are in the chromosome centers. The really important genes, the highly conserved genes, are even more likely to be in the centers. If a gene is in a chromosome arm, it's more likely to be worm, unique to worms, not conserved among animals or eukaryotes or whatever, right? So the centers are gene rich and the arms are gene poor, but the centers have much lower rate of recombination and the arms have a higher rate of recombination. And this is very different from most other organisms. We humans, most of our genes are in regions of high recombination. Fruit flies, most of their genes are in regions of high recombination. Almost all organisms have most of their genes in regions of high recombination, and regions of low recombination are filled up with transposable elements and other crap. Worms are the opposite. And I think that's a really interesting fact. And I don't have it probably has something to do with self-fertilization. Yes. It probably does. But that is also, as far as I know, that's also true of other Caenorhabditis that are outcrossing, that are not selfers. And at least in Caenorhabditis outcross, so gonochorism, so males and females, daiichi, is the ancestral character state. That's another, I could have talked, I could give like a 2-hour lecture and I would have talked about the phylogeny of Caenorhabditis or something. But this self-fertile hermaphroditism, this is a derived character. So the closest relatives of C. elegans are outcrossers. Most Caenorhabditis are outcrossers. So, and yet, I'm pretty sure all Caenorhabditis that anybody's looked at have this similar, centers and arms. Now, we don't have such a good estimate of recombination rate for anything besides elegans and Briggsy, which is also a selfer. So I would say it seems, but then if I step back and think about it, it's like, well, maybe not. Maybe it's not just the selfing. So, but the point being, I think this is interesting, and somebody should figure out what's going on here. The other thing I'll say, if you have taken, undergraduate genetics, you probably learned the lac operon in bacteria, right? So bacterial genes, so prokaryotes very often have genes in operons, which mean basically an operon means that 2 or more genes are co-transcribed under the control of the same promoter. And that's rare in eukaryotes and multicellular organisms, but the C. elegans genome actually has quite a number of operons. And you can speculate that maybe in an organism that's so small with such a fast life cycle, maybe there's a big advantage to having the same set of genes under the control of the same promoter. But that's idle speculation. Okay, so RNA interference, RNAi. So this is the third worm Nobel Prize. So this is Andy Fire and Craig Mello. So it had been known, so this is circa late 90s, and it had been known that if you inject single-stranded RNA, so if you inject RNA into an organism or a cell, right, very often you can screw up the expression of the gene. But people didn't understand what was, it wasn't clear why that was. So what these guys did is they had some elaborate reasoning that I just looked up yesterday for why they thought maybe that, if you inject a lot of single-stranded RNA, occasionally it'll fold back and you get some double-stranded stuff, and maybe that has something to do with it. So they did an experiment. So the sense strand, that's the coding strand, and antisense is the template strand, I think. So basically they've got some gene, right? And this was myosin, if I remember right. So they injected one single strand of RNA to one strand, and basically the worm is fine. You get a wild type worm. And then they inject the other strand into it, and the worm is fine, and you get a wild type worm. But they injected double-stranded RNA, and now you get a worm with it. Basically, it's uncoordinated. So the worm is twitching, and they know they know this is a mutant, and mutant of this myosin gene, and they know if you have a mutant of this gene, this worm will twitch. Right? So they said there's something about this double-stranded RNA that is knocking out the function of the gene. So then they went on to investigate further, and sort of here is, this is taken from their synopsis of the Fire and Mello Nobel Prize. So the mechanism for RNAi, right, is you have double-stranded RNA, and then there is a protein called dicer that chops up, the eponymously named dicer chops up this double-stranded RNA into smaller pieces. One of the RNA strands is loaded into this other protein complex called RISC, and I didn't bother looking up what that stands for. And then this small piece of RNA guides the RISC complex to the complementary mRNA, the messenger RNA for the gene. And this is a signal to destroy that mRNA, so no protein is translated. And so this is, this works in the, in the animal itself, and also it carries over, often it carries over into the offspring. Okay, so this is the Nobel Committee for Physiology or Medicine. So several processes in the cell use RNAi. So double-stranded RNA, there are many viruses, double-stranded RNA viruses that inject double-stranded RNA in the cell. Presumably this RNAi mechanism evolved as a defense against viruses and or retrotransposons. But also the mechanism has been co-opted for a lot of important, it has important functions in the development of the organism as well. So that was, let's see, okay. All right, so the last thing I want to talk about before we just open it up. So this is an experiment from Oded Rakavi, who is one of the more creative and interesting biologists out there. And these are people in his lab, and Oliver Hobart, I think, was his postdoc advisor. So what I have shown, this represents Lamarckian evolution. So you guys remember Lamarck? He's the guy that was wrong, right? And his example, remember the giraffes, and they stretch the giraffes and they get the little tasty acacia leaves at the very tip top. And by this act of stretching the vital force or whatever, I can't remember, he called it, he had a name for it in French, the offspring of this giraffe born with slightly longer necks, right? And inheritance of acquired characters. And never mind that Darwin also believed in the inheritance of acquired characters. But what this shows you here So this is animals with a starvation history show an extended lifespan. So what they did, so N2, this is the worm, this is the control. This is the lifespan of worms. So these would be the great grandparents, right? So some are fed and some are not. And then if the grandparents are not fed, so now the offspring are put back in food. I'll show you the oops. Here's the experiment. So they have starved, so the parents are starved or not or fed, right? And then they get the offspring, and now everybody is all the kids and the grandkids and the great grandkids are all fed. It was only the great grandparents that were, some were fed and some were starved and some were not, right? So if you go back here, if you look at the lifespan, so the comparison of lifespan of worms whose great grandparents had experienced starvation at the first larval stage versus animals whose great grandparents were continuously fed, what you see, so these are the starved great grandchildren of starved great grandparents, they live longer. They have a longer lifespan than the great grandchildren of great grandparents who were continuously fed. So whatever it was heritable over, into the F3 generation. And the F3 is actually key because those individuals there could be no carryover in utero or in grand utero, right? So what's the mechanism? So the mechanism turns out to be something, basically it's an RNAi-like phenomenon. So this is the small RNAs that target a particular gene, and this is the ST gene, but there's a lot of, this is just one representative gene. So these are the genes expressed in the parental generation that are fed. And these are the same genes in the parental generation of the starved worms. And you can see there's a lot more small RNAs in the starved worms. And then if you look at the F3 of the starved worms, there's still a lot more of those small RNAs than there were. And they don't show it here, but the F3s of the fed worms would basically look like that. So this is something I'm really, really interested in. And if I can ever get a grant to do this work, this is what I'd love to do. There's lots of guys working on it, and a lot of them are smarter than me. But okay, so I think that's what I got. So at this point, I would say if anybody hasn't seen the worms and want to, go up on and take a look, and I'm happy to stay here and answer questions as long as you feel like.
Okay, great. Well, thank you very much. Yeah, very interesting. Are there questions here in the room? No?
No, those are just wild type N2 worms. There's no stains in those. The worms in my microscope, you mean? No, those are just wild type worms. I didn't notice that they were pink. Yeah. Wait. There is red ink on the plate, and you were probably looking in the area in which there was red ink. Would be my best guess. I'm sorry. You got a question?
All right. Well, for over 20 years, I did water sampling at Whitney Lab, and that was a weekly sample with water from the estuary. And during those samples, I found maybe 3 to 5 nematodes in there, and I was overwhelmed when I saw that first plate of yours with all those worms in it.
So you were sampling the water column, right?
Yeah.
So clearly, the nematodes don't usually live in the water column. Now, I bet if you were to sample the detritus or scrape some aquatic vegetation, I bet you turn up some nematodes.
Well, we used a plankton net, and that there was a 3-minute tow. That's what we
Yeah.
did.
I mean, so, I mean, I would not predict. So nematodes are bacterivorous, right? So C. elegans is typically found in rotting vegetation, and they eat the bacteria that grow on the rotting vegetation. So if there's nothing for them to, most nematodes, well, there's parasitic nematodes too. Like if you were to catch a redfish or a black drum and fillet it along the lateral line, along the spine, you'll find like little white worms. Those are nematodes that are parasitizing those. So, but there's lots of parasitic nematodes. We know that. I doubt there's the water column would seem to be like the place you'd maybe be least likely to find a nematode.
Well, we only went like 1 to 2 meters deep.
Yeah, I mean, again, my guess would be if you stir up the muck at the bottom, you'd be more likely to find them.
Okay.
So a friend of mine has found them in the Great Salt Lake, and they're, of course, in the detritus.
Okay.
But
And do you work with anybody at Whitney Lab?
not. I have friends and colleagues there. I don't directly collaborate with anybody at Whitney, but I know Joe Ryan. I know Mark Martindale. I just inherited a graduate student from Elaine Seaver. So.
I know both of them.
Yeah.
Thank you, Charlie. I have a question.
Sure.
I do better with concrete examples. What kind of research are you doing that uses nematodes? Maybe you can
Okay.
describe.
So as Jane alluded to, so I am a population geneticist, and I'm interested in genetic variation, and in particular, I'm interested in mutations. So my reason for using the worms was the very first reason that Brenner gave, which is they have a fast generation time, and also because you can cryopreserve them. So if you want to get faster than 3.5 days or 3 days at 25 degrees, you have to go to yeast, and I don't work with yeast. I'm not a microbiologist, so I do these experiments. We call them mutation accumulation experiments. So we get a single worm. It's nearly homozygous, and we inbreed it as much, and then we grow a big population. We start a bunch of replicate populations. We had a board. I could go to the board, I suppose. And then I freeze down the ancestor, and I propagate the descendant populations for a long time, and mutations happen. Every round of DNA replication, there's some probability of mutation, and there's always some probability of DNA damage just to standing DNA. So in the first experiment I did on this, we took them out for 250 generations. So every 4th day, I picked a worm, 600 samples, and then I pull the ancestor out of the freezer, and I want to compare the descendants to the unmutated ancestor, presumably unmutated ancestor. So we can either, if I'm interested in what's the effect of mutation on behavior, let's say. So, and we just had me and my French colleagues just had a paper on this. So they did an experiment. They have a behavioral assay. They ask a very simple question of the worms. In any unit of time, are you moving forward? Are you moving backward? Are you turning left? Are you turning right? Or are you sitting still? Or simple behavior. So we asked them, okay, after some, so this is we've got six left, right, sit still for 5 characters, right? And then the covariances between them. So we ask, after 250 or so generations of mutation, how have these behavioral traits changed? And on average, the mutations slow the worm down. They're more likely to sit still. And which is not surprising. Most mutations are bad. So, and they make just more sluggish. But also they're more, if I remember right, they're more likely to go to reverse course, to go back. Like it ruins their memory. They forget where they were going and they have to go back. And I don't remember what left or right, but definitely the behavior that average, there's a couple of changes in the average behavior, and then the variance increases. So we ask how much genetic variation does mutation put in any generation? And that's the kind of experiment we would do to ask that question. Then you ask, well, who cares? And I would say, okay, I'm an evolutionary biologist. What I'm really interested in is natural selection. So if I go out in nature and collect a bunch of wild isolates of worms and say, okay, now what is the behavior, the average behavior and the variance and the covariance of the behavior of worms in nature? So I could say, okay, let's take, let's say nature wants you to do whatever this is, nature wants you to do this. And then if I look at mutation, these mutation accumulation lines, I say, okay, on average, the mutation accumulation lines do this. So now I say, okay, what is, but, the wild worms do this. Mutations making them do this. Presumably the difference between the two is due to natural selection. And, I picked pointing in this way is like an extremely arbitrary trait versus pointing here. You look at body size, on average, mutations make the worm smaller. That's inbreeding depression by any other name. And we can sequence the worms and say, okay, what is the genomic mutation rate? What is the molecular spectrum of mutations? How does that differ between genotypes, or does it? So those are the kind of questions that I'm interested in. No, these are mutations as God deals them out. I'm not doing mutant screens. I'm just waiting.
I was going to ask you what they do in nature, but you answered that spontaneously.
Oh, you mean
what the worms are doing in their natural habitat?
It's like just snorkeling up bacteria until they've eaten it all. Then they dower. The dowers actually do something interesting. They do what's called nictating, and they will sort of stand up on their tail and wave around. They'll come up to like the tallest thing and hand tall relative to the worm. They crawl up on it and they wave around. Presumably they're waiting for a bug or something to come along, and then they can, a lot of times people would find them associated with isopods, roly-polies, right? That are also rooting around in the muck, snorkeling up fungi and whatever they eat. Some people think there's like a phoretic relationship. There's probably not. It's probably just accident that you just happen to find one on a roly-poly because they were just down in the same. But if you do the experiment, I would say, throw down a banana peel or just go find some rotting leaf litter. Get a plate like this. Throw that leaf litter and rotting vegetation on there. Come back a couple days later. I promise you'll see nematode tracks on it.
And there are lots of different species that are doing similar things, or
yes. And I think nematodes are probably one of the more undersampled of eukaryotic organisms, I bet. These are terrestrial. Well, they're rotting leaf litter, right? But you can. I'm sure if you find it, take a little drop of water on a rotting leaf or something, there'll be nematodes swimming around in it. So it's like their environment can be locally wet, but you're not going to find them. Clearly, you don't find very many of them in the water column. In terms of, yeah. So are these multicellular plankton, or I guess plankton are multicellular? In one drop? Okay. So like cladocerans or other small, rotifers, that sort of thing. Rotifers are super cool too. Yeah.
Well, I think we have reached our we've reached our 2:30 time frame, and I want to thank Charlie very much. This was just perfect for learning about model organisms. Thank you so much.